So we will have today we will have 4 presenters, and Young-Hoon will start with the protein vaccine, and then Sung-Hoon will talk about the DNA RNA vaccine, and then Taek-Chun will talk about the small molecule drugs, and Se-Jin will finish with upcomer diagnosis. Okay, we go with this order. So without a delay, let's just start. Everybody will have a maximum 20 minutes. Okay, let's just start, Young-Hoon.
Hello, today I will gonna talk about the protein-based vaccine for COVID-19. In my opinion, I think vaccine is the fundamental technology to fight against COVID epidemic or pandemic like COVID-19. And for this semester, after the midterm exam, we mainly learned about the immune system. So I think this will be the good material to talk about. You may all read this article from the materials that professor gave us. In this article, there is a statement that one research organization in our country developed a candidate for the protein-based vaccine, and they sold this technology to the pharmaceutical company named Hubei Bio. Like this, fever for the development of vaccines against COVID-19 is very high. So this will be the good topic. Firstly, let me talk about the protein-based vaccine. Virus is usually composed of genetic material and some proteins such as capsid or spike protein. And the proteins that exist on the virus can act as an antigen in human's body like a spike protein. So some recombinant proteins can be vaccines, and that is protein-based vaccines. Sometimes just proteins can act as protein vaccine, or some named virus-like particles also act as vaccines. Virus-like particles refer to particles which don't have just genetic material compared to original virus. And let me talk about the manufacturing process. Usually this is done by genetic recombination. First of all, the portion of the genetic material which produce the target protein is gotten from the original virus, and then that part is injected into bacterial plasmid DNA or the virus DNA, and then that recombinant DNA is again injected into like some cell like yeast cell, which can massively produce the target protein that we want. So this target protein is massively produced, and then scientists or researchers adequately manipulate it and then create vaccines. However, this protein-based vaccine, so-called subunit vaccine, is less effective compared to live attenuated vaccine. A live attenuated vaccine is originally the weakened version of a virus. So they actually in humans body can grow and multiply, but for the protein-based vaccine or subunit vaccine, they cannot do those tasks because they don't have genetic materials. So it is less effective compared with live attenuated vaccine. So protein vaccine vaccine adjuvant is developed in this context. It boosts the immune system and vaccines effectiveness in several ways, and this trade is very good for certain pandemic such as COVID-19. This adjuvant can help vaccine in ways such as making antigens stay in vessels longer or human body, helping antigen-presenting cells absorb antigens easily, and activating innate immunity. and also helping distribution of cytokines. So, and let me talk about the one of the most developed protein-based vaccine against COVID-19. This vaccine's name is MBX-CoV2373. This is on development now by the pharmaceutical company named Novavax, and they chose the spike protein on the capsid of the COVID-19 as their vaccine. This spike protein will act as antigen in human's body and elicit antibody response. So this MBX-CoV2373 is manufactured firstly by getting the portion of the genetic material of the COVID-19 which produce the spike protein, and then that part is injected into baculovirus DNA, and then they infected SF9 insect cell which they discovered, and by infected, they made a system which can massively produce the spike protein, and by this way, they made vaccines. And this is also the adjuvant that they originally developed at maybe several years ago, and this adjuvant name is Matrix-MTM. It is saponin-based adjuvant, and saponins, which are storage or triterpenoid glycosides, are mixed with synthetic cholesterol and phospholipid. This adjuvant stimulates the entry of antigen-presenting cells into the injection site by the increase of release of cytokine and enhances antigen presentation in the local lymph node. This is possible because, as I said on the vaccine adjuvant slide, adjuvant usually make the antigens stay longer in human's body, and they do this by the adjuvant adsorted antigens on their surface. So this is possible, and this effect is also called the default effect. By this way, they enhance this antigen presentation, and they can make them to reach to local lymph nodes where there are many T lymphocytes. By this way, they can boost cell-mediated immune system and antibody-mediated immune system. And so they cleared their preclinical test with good data on baboons and mice, and they now proceed on clinical tests. For phase 1, they diversified the method of injection, and this will produce during about 20 days, and subjects will be treated differently in the volume of vaccine or the existence of adjuvant. And for phase 1, 1 to 130 subjects will be was collected, and their age is diverse from 18 years old to 59 years old. And after that, after phase 1 trial, and phase 1 trial is estimated to end in July, and this means the data is also the data means the collected data, and phase 2 trial will begin after that. And for phase 2, 2,200 subjects will be collected, and more than 1,000 subjects will be elder adults. And they expect all clinical tests should end within this year, and they think this is possible because it's emergency situation, and in my opinion, they get grants from governmental organizations, so they think this is possible. I think vaccine is maybe the most fundamental way to fight against the COVID-19, which we don't have to wait until the COVID-19 and naturally. So this will be the good way to fight against the COVID-19, in my opinion. Thank you for listening.
Okay, thank you, Young-won. Questions for Young-won?
Can I ask one question? I didn't quite catch the part when you said the protein-based vaccine is less effective than the live attenuated virus. So can you explain the reason why it is?
Live attenuated vaccine is originally the weakened version of virus. So weakened virus is just virus, so they can multiply or grow in human's body, and that means there can be many antigens which can be utilized in the immune system. But for these subunit vaccines, they don't have genetic material, so they can multiply or grow in human's body. So maybe in 볼륨 양적인 측면으로 antigens, it will be more effective in live attenuated version of vaccine, and also live attenuated vaccine can boost cell-mediated immune system, which means the innate immune system in our body. But for the subunit vaccine, that process is not occurred or much, so adjuvant is used to be.
Thank you.
Young-won, you're mentioning two different effects. One is attenuated the vaccine. Virus can have many different kind of antigen. That
Yes.
implies that the recombinant the vaccine is just a single protein.
Single protein or virus-like particle, but maybe for the virus-like particle, they also don't have genetic material, so they can't multiply or grow, maybe.
A little different concepts are mixed up here. Attenuated virus also is most likely the DNA or RNA RNAs are destroyed. That's, if not, it can be very dangerous. We don't want to inject the live the virus DNA RNA to the body, then it can make a real virus. So one possibility is maybe the attenuated virus is still, it has every protein on the surface. So it may have a different kind of antigens. So many chances to boost the immune in different ways. Recombinant, usually you choose one protein. You don't use the whole thing. So you said quantitative, sounds like it's more like a qualitative or diversity issues. That may be one possible reason. The other one, you said, whether you need to use an adjuvant or not. So that can be another important point. If it's a simple single protein, then maybe the uptake of that protein into our immune cells, like antigen presenting cells, may not be efficient. So that may be another reason. By the way, what is an adjuvant? What is the meaning of an adjuvant?
보조제.
So it's kind of a helper.
Yes.
Okay. The helping mechanism, you said two things, but basically it helps to observe those protein into antigen presenting cells.
Yes.
If they don't take it up, then they cannot present that antigen. That's the issue. Now, as an example, you mentioned, can you go to the adjuvant, the structure? Adjuvant page. Here, you read Matrix-MTM. What is a TM? Anybody knows TM? It's not transmembrane.
상표 이준입?
In English, what is the 상표 in English?
Trademark?
Trademark. Yes. So TM is a trademark. So you don't want to read the TM. That means Matrix-M is the company's name. Okay. You don't need to read the TM. TM is just showing that it's not component name. It's kind of a company's the product name. Okay? Now, this Matrix-M is kind of a saponin-based adjuvant. What is a saponin? You explained something, but saponin is more famous. Everybody knows the saponin. Where did you hear saponin?
You mean in my life or?
Okay, you don't answer. Other people, saponin. Have you heard of saponin? Saponin is a component of what? It's a famous one, saponin. Nobody knows? Youngwon?
It is in the plant.
Which plant?
Potato?
No,
진생.
Korea's famous ginseng.
Ah,
Saponin is in ginseng. Ginsenoside, those are the class of the representative compounds in ginseng, in some. That is a saponin. So saponin is kind of a detergent, like the soap. So that's
yes.
how it can help
Emulsion.
take up the protein into the antigen presenting cells. So it's kind of a healthy, saponin is kind of a healthy food. So it may not be toxic. If you add it together, saponin, it's like insamgaru together with this the antigen. So it may not be harmful. So then it can help the immune boosting. Even we hear that the ginseng itself is kind of immune boosting the material. So anyway, by some other effect, it can help too. Okay, other question to the Youngwon. By the way, Youngwon, you started with a Korean company name, but this product is not Korean companies, is it?
Yes, I chose this because it is the most fast or the most developed
advanced.
one, the most advanced. Yes,
So you believe this will be the #1 vaccine in the world?
I think as I know, the protein-based vaccine is now usually on the development of for COVID-19 vaccine, and COVID-19 is the thing that people's understanding is less enough. So live attenuated version is too dangerous. So subunit vaccine is the most effective and safe way to against COVID-19
Well,
vaccine.
maybe safer, but you are not sure whether it's effective. That's why it's not effective enough. That's why you need to add helper, so-called adjuvant.
Yes.
Safety-wise, yes, definitely it will be the safer. But whether it's as good as the attenuated virus is not clear. Okay, any other question to Youngwon? If not, thank you, Youngwon. It was quite good. And let's move to the second talk by Seunghan, DNA RNA vaccines. Youngwon, would you take down your file?
Yes.
Okay, you can see the file. Seunghan, you can go.
Okay. Today I will introduce about DNA and RNA vaccine for solution of COVID-19 situation. And this is my content. Firstly, I will introduce about DNA plasmid vaccine and mRNA vaccine and the encoded antigen for this genetic vaccine. And nextly, I will suggest the advantages of genetic vaccine. And lastly, I will introduce the method to commercialize genetic vaccine. DNA plasmid vaccine is a genetically engineered plasmid containing the DNA sequence encoding the antigen or pathogen which we want to target. So we produce this plasmid and inject this DNA plasmid to body. And the company Inovio in US entered the phase COVID-19 vaccine INO 4800 on April 3, 2020. There are two T-cell activation mechanisms. The first one is direct route. When the DNA vaccine is entered to the host cell, like myocyte or APC, this cell synthesizes the antigen, which encoded in DNA plasmid, and these antigens are directly presented to T cell. And the second way is indirect route. In this case, myocyte uptake this DNA plasmid, and myocyte synthesize DNA plus antigen encoded in DNA plasmid. And this myocyte goes apoptosis, then antigens are released out of the cell. Then APC can engulf these antigens, and APC present antigens to T cell. By this way, immune cell responses is activated. mRNA vaccine has almost similar ways to activate immune system. And this mRNA is encoded on antigen of pathogen 2. In COVID-19 case, the S protein in the prefusion conformation before the virus and host cells is the encoded protein of the mRNA vaccine. And the Moderna has begun a phase 1 study on this mRNA vaccine as a vaccine for COVID-19. This is some detail of the antigen presenting mechanism. In briefly, when this mRNA is entered to cell, ribosomes synthesize antigen, and they are presented by MHC class 1 and MHC class 2, and they are activate T cells, CD8 plus and CD4 plus respectively. There are several differences between DNA vaccine and RNA vaccine. Firstly, DNA vaccines should go transcription phase before mRNA translate to protein, but mRNA don't need this transcription phase, and it translates to protein directly. And RNA is less stable than DNA because of extracellular ribonuclease. This is the weak point of RNA, but these two of RNA, so mRNA vaccines are researched by scientists. The first one is uptake ability to nucleus. Some studies show that mRNA can be uptaken to nucleus without any specific uptake system, unlike DNA. And second is interaction with our own genome, like recombination with DNA. So RNA is more stable than DNA in the nucleus of our cells. In COVID-19, encoded antigen will be the S protein because, as you see in this picture, S protein is the biggest and significant protein. So I think the reason that companies are encoded antigen. Here are some advantages of genetic vaccine compared to conventional vaccine or other treatment. Firstly, genetic vaccine has no risk for infection. As Young-hoon said previously, convenience vaccine has a risk
Recombinant
for
vaccine. Recombinant.
a recombinant vaccine has risk for infection, but genetic vaccine has no risk for infection because it encodes only the part of virus like S protein. And COVID-19 is more fatal to elderly people or underlying disease patients, and if someone someone infected by COVID-19 once, they will get destruction of lungs. So the genetic vaccine is a good approach to overcome the COVID-19 situation. And the second advantage is these genetic vaccines can present antigens by both MHC class 1 and MHC class 2. Vaccines only can present antigen by MHC class only CD4 plus T cell, but genetic vaccines can activate both ways of immune cell response. So it is the good advantages of genetic vaccine. It also make immune response focused on antigen of interest, and ease of development and production and stability for storage and shipping is the advantages of genetic vaccine compared to recombinant vaccine or protein vaccine. Then how can you commercialize these genetic vaccines? In several clinical trials, we have some results that show level of immune responses induced by genetic vaccines. So we should optimize immunogenicity of genetic vaccines to commercialize. This vaccine deliver optimized immunogenicity of genetic vaccine. These three are decreasing factors of vaccine delivery. First is rapid degradation of DNA, and second is inefficient uptake of DNA by cell. And the third is inefficient uptake of DNA by nucleus. So we should improve vaccine delivery by improve this
Someone, I cannot hear you. Someone?
One technique of delivery of drug, and they shield the DNA vaccine for degrade.
Maybe someone's the internet is a little unstable. Someone, would you share your file again?
Okay. Can you see my screen?
Yes.
Yes. So we can use this nanocarrier technique to improve delivery of DNA vaccines. And second way is DNA delivery facilitation. Electroporation is the one way of this. We should uptake genes into the cell. So we apply the electric pulse, then pulses are formed in the cell membrane, and the gene-like DNA plasmid can enter into the cell. So after the electric pulse, the pore is disappeared and the gene remains in the cell. The third way is the co-administration of adjuvants to improve immune response. As Young-Woon already said in his presentation, adjuvants enhance antigen presenting response. So if DNA vaccines co-administer with plasmid that encode co-stimulatory receptors in transfected APC, T-cell stimulatory capacity can be enhanced. CD80 and CD86 are examples of these co-stimulatory receptors that can activate antigen presenting responses. So if we optimize genetic vaccines by this method, we can develop genetic vaccines and overcome COVID-19 situation. Thank you for listening.
Okay, thank you, someone. Question to someone?
Yeah, I have a question.
Go ahead.
So, how do we deliver the genetic material into the location we want to send? Is it just random, or we can designate the place?
Oh, sorry, I couldn't hear your voice.
How can you
Tell me
deliver
once
those DNA vaccine or RNA vaccine to desired place? And,
actually, I don't know why we target this myocyte, but I saw that we inject DNA or RNA vaccines into the muscles and we target myocyte.
Okay, myocyte is a cell of a muscle. That means the vaccine was injected into the muscle. So for the injection of vaccine, if you recall your vaccine, the shot you got so far, where did you get the vaccines? Whatever the vaccine you had so far. Influenza vaccine or 불주사 or any vaccine, where did you get? Do you get it into intravenous, into the blood vessel? If not, where? So since the question is, the maybe fundamental question you should ask first, where can be ideal place of vaccine entry?
Actually, I don't know where.
So usually the vaccine is given under the skin, subcutaneously, or into the muscle. So muscle is holding those material longer time and slowly releasing them. So we don't inject the vaccine directly into the bloodstream. That's too abrupt. We want just to keep those foreign material slowly released to our body and reduce the risk and let the other cells represent those antigen as acting as APC, antigen presenting cell. So in this case, myocyte act as APC. If not, maybe then the dendritic cell or macrophages, the other cells, they swallow this DNA or RNA. They make those products. So eventually those antigen is not DNA or is not RNA. Antigen is still proteins, but those proteins are synthesized inside these cells. So vaccine case, actually you don't need it to be specific. Just 대충 inject into the muscle, that works. If that is the question. Now, inside the cell, whether it can be transferred into the nucleus or not. So as someone mentioned, there is some difficulties of the uptake of RNA. RNA may be easier to penetrate through the nucleus compared to DNA. So there's some the intracellular process you may need to concern. But body size is not a problem. Now, one comment the someone made. DNA is more stable than RNA is more stable than DNA in the nucleus. Can you say so? RNA is more stable than DNA in the nucleus.
No, in nucleus is
No. Someone mentioned that RNA is less stable than DNA because of the nucleic the breaking the enzyme. But not only enzyme reactions, RNA is chemically maybe million times or billion times more unstable, less stable than DNA because of the chemical structure with the extra hydroxyl group on 3 positions. So uptake may be better, but stability never. RNA is much, much less stable than DNA. Another point that should be corrected is recombinant. The recombinant protein can infect not DNA, RNA. That's wrong. Recombinant protein is also a single protein, and which is designed. So if it's attenuated virus, if it's less attenuated, yes, it is dangerous. But recombinant protein or DNA or RNA should be same in terms of safety from the infections. They should not infect the bodies. Okay, other question to someone for DNA, RNA the vaccine? One question here. What did you say? If you use the protein vaccine, it can use MHC 1 or 2. Which one? You mentioned using this picture. DNA vaccine, genetic vaccine can utilize both MHC 1 and 2. If it's a protein vaccine, You said only one can be used. Which one can be used?
Class 2.
Protein vaccine only can use the MHC class 2, is it? You said something, someone. What did you say?
I said protein vaccine use only MHC class 2.
Okay, find that reference or the material and send me separately later. Okay. Okay, we don't have a time, so we should move on. Let's move to the next the talk. It's section. Section is going to talk about the small molecule drugs for fighting against coronavirus.
Can you hear me well?
Okay, very good.
Okay. I will introduce about recent research about small molecule drugs. So I introduced about what kinds of candidate small molecules exist and recent clinical trial I will introduce about. So as you can see this picture, there are several strategies to defense against SARS coronavirus 2. There is a strategy that interfere the entry of the virus and interfere the degradation of virus's protein and or interfere the replication of virus. The first small molecule is remdesivir, is the inhibitor of RNA dependence, RNA polymerase. And this drug was developed by Gilead Science. And in the recent clinical trial, this drug was used to 53 patients. And the result is clinical improvement has shown in 33 patients. So it might seem this drug is quite effective. But interestingly, patients who have worse symptoms fail to see the effect of this drug. So it means this drug is effective for mild symptoms or moderate symptoms patients and not good for worse symptoms patients. And also this drug has some adverse effects such as renal impairment or abnormal liver function and hypertension. So further study should ongoing. The second one is Caletra. This drug is inhibitor of protease, so it interferes the degradation of viruses protein. And in clinical trial, this drug was used to 200 patients compared to the control group. And after the 9 days of treatment, there is no clinical improvement, and both groups have similar mortality rate. So this result says that Caletra don't have any clinical improvement, but number of patients who have secondary infection or requiring mechanical ventilation for respiratory failure, the number of patients were fewer in former group who have used Caletra than control group. So this means additional research required to determine whether this drug can reduce complications of COVID-19 treatment. The third one is Umifenovir. This drug interferes coronavirus's spike proteins and membrane fusion. So this drug can inhibit the entry of coronavirus. And there is two clinical trials. The first one, 33 patients received these drugs compared to the control group. And the result said this Umifenovir decreased mortality rate and reduced viral load determined by RT-PCR. I slightly understand that viral load means how many viruses can detect in body fluid. And second clinical trial, 16 patients received both Umifenovir and Kaletra, both drugs together, and 17 patients received only Kaletra. And then result used both Umifenovir and Kaletra have good result. Specifically, patients have chest computed tomography scans. It is CT 촬영. After these patients have the CT 촬영, CT scans. Patients who have Umifenovir and Kaletra, both drugs, were improving in over 70% of the groups. So more clinical trials with this Umifenovir combination with other drugs such as Kaletra and Chloroquine have been recently initiated in China. And the final stat, this drug is inhibitors of TMPRSS2. This is a transmembrane protease series of family member 2. And this protein is associated with entry of coronavirus. It is associated with spike proteins and ACE2 enzyme. This drug is already approved drugs by FDA. This drug is used for MERS. So this drug was already approved FDA drugs. Since the spike proteins of MERS and current coronavirus share considerably amino acid sequence, so this drug can inhibit the entry of SARS-CoV-2. So there is evidence that this drug can inhibit SARS-CoV-2's entry into host cells with roughly 15-fold higher efficiency than camostat mesylate. Camostat mesylate is another candidate drug, inhibitors of transmembrane protease, but the famostat is more effective than camostat mesylate in low nanomolar range. And chloroquine and hydroxychloroquine is fairly familiar drugs. We have already seen this drug in the article. And there is evidence that chloroquine inhibits entry of coronavirus and cell fusion, and hydroxychloroquine can reduce cytokine storm and can reduce symptoms through its anti-inflammatory properties. And I found that there is some interesting research that zinc enhances the chloroquine intracellular uptake. So further study combining zinc with these chloroquine and hydroxychloroquine are currently under investigation. And moreover, another studies are ongoing to evaluate the safety of these drugs, but it seems a little hard because many other studies shows that high dose of these drugs is associated with severe cardiac risks. So treatment of this drug is still controversial. And azithromycin is also have anti-inflammatory properties. So this drug has been used as anti-inflammatory effects for treatment. And currently, many trials are testing the effect of this azithromycin combination with hydroxychloroquine. And the result shows that the effectiveness is better than use only hydroxychloroquine. But the mechanism of azithromycin, how azithromycin enhances the efficacy is unclear. So further study should be prove the mechanism. And this is I search about candidate small molecule drugs and clinical trials, and as I saw the newspaper, Chinese scientists have found another protease inhibitor drugs. So I will introduce about that. First, Chinese scientists determined the crystal structure of protease, and then they determined the structure of protease complex with inhibitor, and then they searched about 10,000 drugs, including approved drugs or candidate drugs in clinical trials, and finally they found 6 molecules role inhibitor of protease effectively.
Actually, you may pronounce this the enzyme name is a protease. Protease.
Okay. Sorry, protease.
That's
So this graph shows 6 molecules of protease inhibitor, and we can see IC50 value of each molecules. And the first one, epsilon, is the most effective molecules to inhibit the protease. So I think the development of this epsilon or further study of development of combination with this drug with other therapies is will be a promising topic. This is my end of presentation. Thank you for listening.
Okay, thank you, Taekjun. The question to Taekjun. Taekjun, the last part of M-Pro. What is M-Pro?
I think this pro is not here. It is pro should be upper subunit. In case of the Young-hoon's presentation in MTM like that, I think this is just yes, kind of that.
This M-Pro is a viral protein or the host protein?
This is viral protein.
M-Pro itself is a protease or M-Pro is a substrate of a protease?
I don't know exactly, but as I read the paper, this M-Pro is associated with this so M-Pro is a main protease of the virus. I looked.
Yes, it is. So M-Pro is a protease of a virus. So if you block the function of the viral protein, the protease, in the beginning you mentioned the protease is breaking down the degraded protein, but actually protease function is rather than degradation. It is a cleave of a multiple protein into each functional protein. So it's kind of more editing protein rather than degradation. It's not breaking enzyme. It is a cleave, but it is actually make those combine the proteins into functional, each individual functional molecules. So that's more like an editing protein. So if you block those virus protease, then virus cannot generate the functional unit of the proteins.
Okay.
Okay, a lot of informations that Taekjun introduced. If we have more time, then each class of compounds we could discuss. It's a little too much. Would you go to the IG50 values, the graph of the last page? Okay, in this page, epsilon is the best compound in terms of efficacy or potency.
Potency. Sorry.
Okay, this is assuming that 100% activity is the same. So that means efficacy is the same. Potency is the best for the epsilon.
Yes.
Okay, thank you, Taekjun. Let's move on to last talk by Sejin Sanbae.
All right, let me start it. So this is Sejin. Today I'm going to talk about a fancy technique, aptamer diagnosis. This is the end of the presentation in this semester. So in March 2020, Professor Cindy Zhang made headlines with the news that his research team developed a diagnostic method to determine whether you were infected with the virus in 15 minutes using a type of molecular capture aptamer. The concept of the aptamer may be unfamiliar to you. This data is on the contents on the Aptamer Sciences homepage, where Professor Zhang is leading as a CSO. So aptamer is a short, single-stranded oligonucleotide molecule consisting of DNA or RNA. Aptamers recognize and bind targets by a three-dimensional structure, while antibodies recognize linear epitopes of the target. In addition, aptamers are highly safe biomaterials with no reported in vivo immunogenicity. It can bind various targets ranging from simple small molecules like peptides to proteins with high specificity and strong binding force. And if you know the sequence, you can synthesize it in large quantities at low production cost, which is considered to replace antibodies. In case of DNA, aptamers are highly stable and easy to transport and store. The aptamer requires a kind of in vitro selection or in vitro evolution to acquire high specificity against target antigens. Aptamers are generated in vivo from an oligonucleotide library containing random sequences by the method named SELEX. SELEX is a biochemical process consisting of repeated rounds of 4 main steps. First, target molecule incubation with aptamer libraries, which containing random sequences and the primer binding site. Second, illusion of bound libraries through the washing. Third, amplification by PCR with polymerase with PCR, and finally, single-stranded oligonucleotide separation. This whole procedure is generally repeated 8 to 15 times, and then analyze the selected aptamer candidates using cloning technique. It seems similar to phage display method I presented last time. It was previous research. However, if the target antigen is a virus, it is not able to produce the proper aptamer through this SELEX because it is difficult to purify the membrane protein, the coat protein used as a target. Instead of purification of membrane proteins, the research team, they combined the baculovirus to make the virus have targeted proteins on its surface. This is a virus SELEX, so-called virus SELEX method. In this picture, the positive selection is to literally select the aptamer candidate that actually bind to the desired target molecule. Negative selection, on the other hand, is the process of removing an aptamer that shows strong binding affinity on proteins that exist on the virus but do not want to combine. So through these processes, the aptamer is selected to have strong binding force and high specificity only to the material that we really want to target. Based on this method, the research team has discovered a new aptamer that acts on the influenza viruses called protein. We already know hemagglutinin. In addition, they succeeded in developing a diagnostic kit that can diagnose virus infections only with changes in color, such as a pregnancy diagnostic kit. Using this device, 50 minutes is enough to check for infection. So before coronavirus, the research for the flu was carried out first. So then, how do we diagnose the disease with this aptamer? Here is a simple diagnostic kit using the hemagglutinin specific aptamer. The kit is composed of a sample pad where the sample is loaded, a conjugated pad containing detection aptamers associated with golden nanoparticles. The golden nanoparticles is kind of stabilizer, and a test line containing a capture aptamer, a control line containing DNAs complementary to the detection aptamer, and finally an adjuvant pad. So principle is like that. If influenza viruses are present in the sample, the detection aptamers associate with the influenza viruses at the conjugate pad and migrate toward the adjuvant pad. The capture aptamers at the test line grab the influenza viruses together with the detection aptamers, resulting in the colorization of the test line. I mean the change of the color at the test line area. The unbounded detection aptamers continue to migrate until they encounter the control line where they associate with the complementary DNAs. So I think my explanation is kind of hard to follow, so I prepare the simple YouTube video. Do you see my YouTube video?
Nope.
No.
Nope. All right, give me a second. I'm gonna change the sharing screen. I think now you see my video, right?
Yes.
All right, so just watch it. All right, come back to my PPT. So the video was pretty much different from this test kit because they used antibody instead of the aptamer, and they targeted two different antigens at the same time, but the principle is quite same. So recently, this technology has been known to be equally applicable to coronaviruses, opening up the possibility of fast diagnosis of COVID-19. Researchers are continuing with the ultimate goal of establishing a platform to rapidly develop diagnostics or even therapeutics for new viruses such as SARS and MERS, as well as COVID-19 virus. So I sincerely hope that the research has done well and help to end this pandemic situation soon. Thank you for listening.
Okay, thank you, Sejin. Any question to Sejin?
Can I ask one question?
Yep.
Go ahead.
Can you explain the principle that how does the change of color has been occur when the test line cut the tumor at the test kit?
Actually, I didn't check how the color was changed after the binding of aptamer. I think that's a good question.
Okay, you
didn't
can see
find out.
those the antibodies bound the red one. So on the tail of the red one, there is a small dots, right? Those small dots can be either fluorescence molecule, or usually it can be golden nanoparticle. So antibody labeled with the golden nanoparticle is kind of a primary antibody labeled with a reporter. So golden nanoparticle can give you strong scattering signal. So either scattering by golden nanoparticle or fluorescence molecule is a common way you can see.
Professor.
Okay, in Sejin's the kit, let's just say the aptamer is binding to which target?
In case of this, or
Professor Seung-Jang's the cases, what is the target of this aptamer? Aptamer binds to what?
spike protein of coronavirus.
Spike protein.
Yep.
Okay, the key idea here, the aptamer is made of DNA.
In this case, they're trying to develop a DNA aptamer. Yeah.
Okay, DNA aptamer made of the DNA, but they are not acting as genetic information. They act as a binding material like antibody. That's the key concept. Okay, antibody is made of a protein, and protein has positively charged, negatively charged, and hydrophobic, hydrophilic, diverse the amino acids, 20 of them. But DNA has relatively homogeneous, all negatively charged, and only ATCG difference. But if they form secondary or tertiary structure, they can act as a binding motif to whatever. But this selects is the designed to select specific binding DNA or specific binding RNA. That's what we call selects, and this is the main the idea of aptamer. We didn't have a chance to discuss fully about this, but at least you should not be confused the difference between DNA vaccine versus DNA aptamer. Okay, so that's most important part. Any other question to Sejin? Okay, Sejin, can you go to the strip information? Okay, that page, follow it up. No, the right page,
This one?
the first page.
Maybe this one?
Yeah, this one. So as you can see here, you can flow those samples from one side to the other. In this case, from left side to right side. There are 3 detecting sites. Usually, it can be either 2 or 3. So one can be negative control, one can be positive control. And in this case, they have maybe the red one is kind of a control, whether the flow is working or not. And then the real experiment, maybe those the green one, you can see the virus is bound, and then it will be the sample test. And blue one can be another control, either just showing positive control or negative control. Kind of things. So very similar concept of like a similar the kit we use the pregnancy the test. In this case, it can be either antibody or it can be either aptamer. So Professor Zhang's case is which stage are they right now? They can make this into product sometime soon?
Actually, as I know, they don't get any items approved by KFDA yet. I think it's still on a research and still on a development for the COVID-19.
Okay, this type of the detection is the target is a protein. So from virus, you can choose one target protein, and either you can use either antibody. If it's antibody, you call this approach as ELISA. Now this is the aptamer approach, very similar, but instead of antibody, you use the aptamer as a binding motif. So currently, so-called the Korea K-kit for coronavirus, all of them are not this kind of a protein binding kits. All of them are RT-PCR based gene amplification method. So this method is much quicker, but most significant problem is the sensitivity. If the number of virus particles is not high enough, then you may lose those signal. Even though there is some virus, but detection limit is not high enough, then you may lose. But in artificial case, you have a chance to amplify the signal by PCR. So that's why so far, none of those, the ELISA-based or this type of protein binding aptamer-based is approved for official use for coronavirus detection yet. So you need to have some other method to amplify the signal. That's kind of a critical issues here. Okay, guys, the quality of presentation was quite good, and I'm very glad to see that in general, your English and also your presentation, the skill was improved quite a lot. So for summarizing of today's the class, actually this is the last class of this the course. Now we'll take the final exam next the Tuesday 2 p.m. and I will use some of this presentation material. I will make some problem out of here too. So and also I found many of you made changes of your files. So if you made any change to your files and send me those updated file right after this class by email, okay? Not to the iClass, by email. I saw some the add up from Younghun and Seunghan, so maybe similar things to the other presenters too. If you changed anything, send me those updated file within 10 minutes of this class, okay? If you didn't change anything, so you just send me message into the Kakao, I didn't change anything, then I will use your old file to upload, okay? That's the one. And for next week, the exam, I will give you the guide by Kakao. So pay attention to Kakao, the Kakao Farm. So by 2 p.m. next the Tuesday, it will be almost the same the format. But anyway, I will give you the guide what to do. So next the Tuesday, we are not going to meet through Zoom. So you just check Kakao, okay? And you confirm that you check those messages. So that will be next week the exam, starting from 2 p.m. until 3:15, exact time of our class time. And so we finished all the presentation today. And this week, iClass, I left one the final assignment there. Maybe some of you already have seen. So if you didn't finish that up, then that is last the assignment. Please upload your last assignment by tomorrow morning 6 a.m. tonight or by tomorrow morning. Okay, so you check 13 the week 13 the iClass. The same week I will put up these files of a presentation file, and you can check those files, and also you submit your the last assignment within today, means by tomorrow morning at 6 a.m. Okay, any questions so far for anything? And I will open up the last week the iClass part, and there is the question on the site. If you wanted to give me any question, and then you can put up any question over there, or you can use the cutoff bound if you want to get the response right away. Okay. If you have any question, then you can give me a question after this class. If not, we communicate again next Tuesday for the final exam.
Thank you,
Okay,
Professor.
thank you guys.